Mrs. Belo here. In this video, I'm going to be going over the murder and a meal lab in our biochemistry unit. You can read what the lab is about more in detail right here, but essentially there was a murder here in Wheaton, in our little town, and we have to solve for where the victim last ate their meal prior to death. And we're going to be using our knowledge of macromolecules to figure this out. Before we get to the lab itself, there are some pre-lab questions that will help with the lab part itself and figuring out where the victim last ate. So for the pre-lab, I have the 3 restaurants where the victim last ate and what they like to eat at that restaurant. Answer the questions that are highlighted in light green, you can see here, as to what macromolecules would be present in the victim's stomach contents if they last ate their meal here. So for example, if one of the places the victim liked to eat was Gino's East Pizza, and they liked to order a thin crust pizza with sausage, pepperoni, and bacon. So based on what they would order, tell me which macromolecules you would expect to be present in that food, which would be in their stomach contents. So that includes carbohydrates, lipids, proteins, and/or nucleic acid. So list all of the macromolecules that you would expect to be present in that. You're going to do the same thing for Buffalo Wild Wings, where they like to eat blazing wings and celery. And then Gia Mia's, where they like to eat bread, pasta, and olive oil. Please make sure that you check your pre-lab questions with me to ensure they are correct, so you can do the rest of the lab correctly. And then also, there's a couple more questions. Answer this one: which one of these meals or meals above would you expect to find nucleic acids? So use your notes to help you with answering some of these questions. Next, you are going to complete this table here. And then you also have question 6, which I'll get to in a second. So this table is going to help us with the lab, and it's going to help us figure out what test are we going to use to test for certain macromolecules. So there are 4 different types of tests that you're going to learn about. There's one for lipids, there's one for proteins, and then there's 2 for carbohydrates, there's 1 for glucose, and 1 for starch. And in question 6, make sure you distinguish between carbohydrates, glucose, and the carbohydrate starch. Again, use your notes to help you. And you're probably wondering, why aren't we testing for macromolecules? For nucleic acids, Because, kind of going back to this question here, well, nucleic acids, DNA, that is present in all living things. So that includes plants, which is where all of this food, plants and animals, where all of our food is coming from. So to answer question number 4, which of these meals? All of them have nucleic acids, because they all came from once living things. So that's why we are not testing for nucleic acids here. So in order to learn about these tests, I have a list of the test here for you. So the first one is actually listed here, and the rest are in the video. So for the lipids test, or the fats test, it's called I'll actually fill this one in. It's called the brown paper towel test, okay? And so you're going to tell me what the test is in the video. For the remaining 3, they are very clear what type of test it is. And then you have to indicate what a positive result would look like, so meaning that macromolecule is present, and what a negative result would look like. if that macromolecule was not present, okay? So for these ones, they might just be like no change, like there's no change in the color for these guys here. That would be acceptable as an answer, or it would remain the same color. Here is where you're going to talk about the positive result, which is usually some type of a change. Maybe it's the change in, for these guys, it has to do with the change in color. But I am going to do the lipid test with you first. So going back to this, so what you do for the lipids test is you use a brown paper towel and you put the substance on the paper towel. Use a permanent marker to divide so you know what section is what. So we use usually water as a control. So we put a drop of the liquid we're testing onto the brown paper towel. And if there is, oh, here it is, this part right here. And if the macromolecule, which is lipids, is present, it will leave a greasy mark, which is translucent, okay? Which makes sense because lipids are fats and they leave a grease stain. So a positive test result would be translucent, a negative test result would be not translucent, okay? So when you hold up that paper towel to the light, if there are lipids present in that solution or whatever it is you're testing, it would be see-through and translucent. If there is no lipids present, it would be not see-through and not translucent. Okay, so you're gonna fill out the rest of this using the video here. Please don't watch the parts of the video that talk about these tests because we don't need to know those. You're gonna figure out which of these three tests test for these guys right here. So there is the iodine test, the Benedict's test, and the burette or biuret test. For the lab itself, if necessary, I will provide you the data, okay, what the test results were. So you're going to kind of repeat what you did for the pre-lab here, the chemical test name, and then this is what the actual results of the test are. And then you're going to tell me, is that a positive or negative test result, and is that macromolecule present or absent? So if you have a positive test result, it would be present. If you have a negative test result, it would be absent. And then here's the data from the lab of the victim's stomach contents. Don't forget to also include a descriptive title for Table 2. So Table 1 here is a really good example. So this says positive and negative results for the presence of organic macromolecules, which you can use something similar for this, but make sure that you also mention that you're testing the victim's stomach contents, okay? So you can kind of use Table 1 as an example, but for your Table 2 title here, make sure you also talk about the fact that you're testing the victim's stomach contents. Someone should be able to read the title of that table and understand what it is that you collected here for your data. Once you have all of your data, you're going to have to think about where the victim last ate. So you're going to have to write a claim. That's just the victim last ate at blank. So again, it's one of these three, and you have the pre-lab filled out here with the correct answers once you've checked them with me. So based on the result, the pre-lab questions here, use that to help you determine where the victim last ate. And then you're going to analyze your results and explain the lab results. So basically you're explaining why that person, why the results confirm where the person last ate. Using your knowledge of macromolecules and talking about the test results here. You should be able to explain it in a way that a person like a police officer that might not have a science background can understand what it is that you're trying to convey. and that is the Murder and a Meal Lab.